Thank you. So thank you for that very nice introduction. It's a great honor and pleasure to be here, and I'd like to thank Professor Wu for the invitation. I'm going to talk about a topic that's been sort of a hobby of mine for the last several years, which I hope is gradually getting more attention from others, though it's been a long struggle. I have no financial interest to declare. Just to tell you where I'm from Nashville, Tennessee, which is that red dot sort of in the middle of the US. And this is the riverfront, and if you come to Nashville, you'll see where all those bright lights are. That's where all the honky-tonks are in the country music scene. And you're welcome to come, and I'll show you a good time. The lower left there is the medical center where I work, and then within that we have a building which is the Institute of Imaging Science, which is about 150 people, faculty, trainees, and staff who work on nothing but advanced imaging methods. So for those of you probably all know this, but MRI has been around now for over 40 years clinically. It records these very nice looking images of what are called the magnetic resonance properties of mainly tissue water, exquisite resolution, a millimeter or even smaller, excellent soft tissue contrast, gray and white matter, widely used in neuroscience as well as clinically. And what you were seeing there over on the left was, I'll play it again, was a fly-through of my or somebody's brain at 7 Tesla, 3D 7 Tesla image at very high resolution. And this is sort of now very well established. Functional MRI, fMRI, is only about 35 years old. It's also well established in both clinical and basic neuroscience, and it relies on detecting very small changes in the brain signal measured by an MRI scanner as a result of changes in neural activity. And in particular, if you do something like tap your fingers and you compare the signals before and after tapping the fingers, what's shown over in red on the slide there are the motor areas, the network of motor areas that are activated by that simple task. So the idea is that you have a stimulus, you get changes in neural activity, you get what's called a hemodynamic response, and that's a word I'll use quite a lot, and we get then a change in the MR signal. We call this the BOLD effect, blood oxygenation level dependent effect, because it's a change in the amount of deoxyhemoglobin in the tissue that drives it. So just to recap how that actually arises, if I have normal tissue, I have metabolism, I use up oxygen, a lot of the blood becomes deoxygenated, and deoxyhemoglobin is paramagnetic. That means it will affect the MR signal. If you change neural activity, you get an inflow of oxygenated blood, the tissue becomes more oxygenated, and the signal goes up. And that's why it's called the BOLD effect. So when you go from baseline to activated, you get an increase in the signal. And it's quite small, but if you do an experiment such as you flash a light for a very short time, you'll see there the transient change in the visual cortex at the back. And if you tell the subject to press a button when they see that change, you'll see that change at the front of, or on the nearer the front of the brain. That's the motor system. So if you map out that signal versus the function of time, you get these curves. The one in blue is from the visual area, the one in red is from the motor area, and that transient change is what we call the hemodynamic response function. And it lasts for a few seconds, and that'll be a very important thing to remember. I'll come back to this later. But you can measure this HRF from a very short transient signal. Now, if you look at the growth of MRI, fMRI, since 1990, there's been over 20,000 publications. Some of them are quite good, some of them are not so good. But if you can see this is an enormous increase in the use of fMRI, particularly in the neurosciences, psychology, and the like. Now, another thing that came along a few years ago is so-called resting state fMRI, in which you don't have to give a task. You don't have to flash the light or press the button. You just have somebody lie there in a so-called resting state. And what you then measure is a whole series of images, maybe a couple hundred or a few hundred. And then you can look at one part of the brain, one voxel or one region, and you can measure its signal versus time. And you can take another part of the brain and measure its signal versus time. And if those two signals are correlated in a statistical sense, then you say they are somehow working together. They're engaged in the same task. And so we talk about things like functional connectivity, which is derived from these correlations of these resting state signals. These signals are very weak, a fraction of 1%. They lie in some range of frequencies up to about 0.1 hertz, so they're kind of very low frequencies. But measuring these correlations has become very, very common in psychiatry and neuroscience because we think they're telling us about networks and circuits. The underlying hypothesis is that if there's hemodynamic synchrony, there must be some kind of neural interaction. So just to recap that, if I take 256 images, and you see there the green blob, the signal might vary. It kind of looks like random, but buried in there may be some information. You take the signal in the blue, and you get a similar trace. You calculate a simple thing like the correlation between them, and if that is significant, above some value, then you say they must be connected. And this is the first paper that described that at the top. That goes back to 1995, Bharat Biswal and others, where they showed on the one slide the result of actually measuring the signal change produced by finger tapping, and on the other, they show the resting state correlations to a region of interest within the motor system. And you'll notice they look very similar, meaning that these neural networks that we derive from a resting state actually reflect those networks which are engaged during a task. And on the bottom there is my first introduction to this field. We did a very simple thing. We looked at a resting state. We took an area in Broca's area on the left, which is this bright patch here, and you say, show me all the areas which are correlated in the resting state, and you see the contralateral area as well as Wernicke's area at the back. So this now dates back almost 25 years to those kinds of studies. Now, this is the growth of resting state fMRI, and you can see that's become a major fraction of what's being done. It's several thousand papers now using this correlation idea. Now, the Bold effect is all concerned with gray matter and the cortex. That's actually a poster for a Bollywood movie. It's not actually about the Bold effect, but it's called Bold. And this is a poster for another movie called Gray Matters. So we have gray matters, we have Bold. Why is there no mention of the white matter? So my outline of the talk is really going to be about demonstrations that the Bold signal in white matter is real, that you can measure it, and there are various metrics I'll introduce, things like the hemodynamic response function, the power spectrum of these low-frequency fluctuations, and a thing called the hemodynamic response function. And I'll try to look at some of the factors that modulate that NMR or that Bold signal in white matter, and even get onto some potential applications, at which point they'll flash that time over sign, and that'll be the end. So the question arises, are there Bold effects in the white matter? There are thousands of reports of using fMRI to measure brain function. Almost none of them, I'll show you a slide in a minute, almost none of them mention the white matter. The white matter is more than half the brain. In fMRI, if you see signal in the white matter, and I'll show you that that's quite possible, people just ignore it. The neuroscience community is not interested in white matter, particularly the psychologists. So if they see that signal, they somehow get rid of it, often by some kind of processing to kind of decrease these number of artifactual things as they see them. And in fact, part of the standard processing in regular fMRI is to measure the signal in the white matter and regress it out as a nuisance regressor. Well, if you regress it out, you're never going to see a signal. Another question is, do we expect there to be a Bold effect in the white matter? And that's sort of unclear because it's not obvious where the energy demand would come from for increasing blood flow, which is what happens in the Bold effect. So we're not even sure if there should be a hemodynamic effect. And in fact, neuroscience has been relatively uninterested. So in here is, again, the slide of functional MRI versus time. And the dark green are all fMRI papers, and the light green are the ones that actually mention white matter. Now, they look similar, but that's a log scale. So of the 18,000 papers that have come out using fMRI, about 400 and some odd actually mention activation in the white matter, and that's sort of somewhat disturbing. And why is that? Well, this is, I think, how people conceive traditionally of white matter. If you think of your metro stops, this is the London Underground, you have the tube lines that go between them, the tracts. They're not very interesting. Everything happens in the stations. Same with a computer network. The cables connecting, the USB connectors or the USB-C connectors connecting things are not that interesting. All the interesting stuff happens in the processors. And this is how people have conceived, I think, of a lot of white matter neurobiology in the past. But that has changed, certainly clinically in recent years. is increasing evidence of involvement of white matter functional integrity or lack of it in various disorders. These are just a few topics I've picked out at random, but changes in the white matter during things like learning, so it's a cognitively involved, how schizophrenia may actually be a derangement of white matter, how Alzheimer's disorder and degenerative diseases may actually originate in the white matter and cause symptoms long before you see things in the cortex. So there is some increasing interest. Not only that, but if we think about the Bold effect, the blood flow in the white matter, even though it's a quarter or a third of the gray matter, their oxygen extraction fractions are actually quite similar. That means there is metabolism going on there. The white matter contains a very different composition to the cortex. There's a much higher ratio of glial cells, things like astrocytes, oligodendrocytes, to neurons. And these astrocytes are also receiving increasing attention. They are known now to perform a number of important functions, things like synaptic modulation, glycogen storage. All the glycogen in the brain is actually in the white matter. Synthesis of neurotransmitters, and also a very important role in some kinds of metabolic recycling. If you actually look at the Bold signal in the white matter, it's not trivial. It's about half or even up to 60% of what it is in gray matter. And we can obviously demonstrate Bold effects in white matter by doing some simple things. So this is a very simple experiment where you take a subject, put them in the magnet, measure their resting state signal, and have them breathe air that's been enriched with 5% CO2. And that's a hypercapnic challenge, and what happens is you cause massive increase in vasodilation and increases in brain blood flow. So it's not an activation task, but it shows you how big an effect you could get if you were to replace a lot of the deoxyhemoglobin with oxygenated blood. And over on one side, you see the effect in the gray matter, and you start breathing as a result of hypercapnia. The signal goes up by 2.36%. That's the Bold effect. In this case, not caused by activation, but by pharmacology. And in the white matter, you see it takes a little bit longer, and it's about half as big. So there's no doubt you can produce a Bold effect in white matter by this means. Also, you can then do simple experiments. So this is an early study done by my colleague Zawad Ding, where we did two acquisitions. First of all, a conventional task where you brush the palm, so you cause sensory stimulation to the hand region. You do that on and off, on and off. And you say, show me the areas that activate or change, and those are the areas shown in red. Now you can also do diffusion imaging, whereby you can look at white matter tracts, and you can say, show me in purple the thalamic tract that connects to the cortex, to the sensory area. And you can then begin to look in the resting state at how does the signal in that white matter tract change when you're doing the task of activating the sensory area. And that's what's shown here. So on the left there is the right palm stimulation, and on the left palm. This is the signal in the gray matter. The red is the task going on, off, and then the signal change is what we measure in those red areas in the sensory cortex. This is what we measure in that thalamo-cortical tract connecting the gray matter to the thalamus. And you can see it's a very robust signal that goes up and down, time-locked, just like the cortex. So this was one of our first ideas, that we could easily get enough signal by averaging over the white matter tract. But in fact, you don't need to do that. You can do it on a single voxel basis if you use the right kind of analysis. So this is another task wherein the task of a visual flashing checkerboard is shown in black, the kind of the square wave. Shown in red and green are the signals from gray matter and white matter, visual cortex and the optic radiations. And if you take the Fourier transform of that, just measure the power spectrum and then say, show me all those voxels that change at the fundamental frequency of the task. This is what you get, and these are the optic radiations deep in the white matter. So we can do this on a single voxel basis and prove that, in fact, there is significant modulation of the signal from the task in the white matter. In fact, it's a little bit cheeky on our part, but this paper came out in PNAS 14 years ago from the NIH group, where they did this massive averaging of very high quality, long duration acquisitions. And their point was, this is just gray matter, their point was that even in a very simple task, such as visual stimulation or motor activation, pretty much everywhere in the brain shows some modulation that is synchronized with the task. And this is their results in PNAS, showing that, in fact, these regions that you don't normally think about as being involved in one of these tasks, actually, they all show some kind of modulation. So we took 3T data. We did about 20 subjects, and we did exactly the same thing, except we looked only at the white matter. And we published our paper with almost an identical title in the same journal, PNAS, showing that, in fact, everywhere in the white matter, these are gray matter, these are white matter, but pretty much in all of these white matter tracts, you can find a time-locked signal just the same as you can for the gray matter. So my point here is not to decry anything else, but whatever you do to look for signals in the gray matter, whatever signal processing you use, you can look and find them also in the white matter. So how do these activations vary with the task? Well, a very common task is to show a checkerboard like that, which reverses, and it flickers at a certain rate. And it's well known that depending on what the rate is, you will get a different degree of activation. So over on here is the signal in V1, the primary visual area, for 3 different flicker rates, 4, 8, and 12 hertz. Then we looked in the optic radiations for 4, 8, and 12 hertz, and you see exactly the same kind of modulation. So that tells me that the magnitude of the signal in the white matter is directly related to the signal that's happening in the cortex. So it's not like a new phenomenon or an additional thing, that in fact, white matter bold signal is driven by neural activation somewhere else in the brain. Now, it turns out, again, this was kind of highly novel at the time, I think, you can find it in the spine too. So the spine has gray and white matter. The spine, the anatomy is reversed, so the gray matter is in the middle, this butterfly shape, and then the white matter is arranged around it in different tracts. And if you do a simple stimulus like a thermal stimulation of the hand, and you look in the gray matter, the ventral horns in the gray matter, you can see those areas that light up in response to the task. This is the signal going on and off, on and off. If you average that over one epoch, you get this hemodynamic response from a single task. That's in the gray matter. If you do the same thing in the white matter, you can get very similar findings, very similar modulation, very similar averaged signal. So this is not just true in the brain. You can find it also in the spine. And then that was all with the task. So then the question arises, do you get resting state functional measurements or functional connectivity in the white matter too? And this was an early result, again, where we simply took a subject, put them in a resting state, acquired a bunch of images, took a seed voxel here, a couple of voxels here, and say, show me all those areas that are correlated in a resting state, and that's the areas shown in red, and clearly that's the optic radiations. So not only can we do a task-evoked signal in the white matter, but white matter shows these resting state behaviors just the same. Well, if they show resting state behaviors just the same, we should start to think about functional connectivity between white matter areas, just like people have used fMRI to look at the cortex. Now, is it the fact that John Gore is so brilliant that he's the only person to have discovered this? I would open that to debate. Or is it because, for some other reason, people have ignored it? And I said before, people don't like to see things they don't expect, and so they can get rid of them. This is actually a teaching paper. This is from the American Journal of Neuroradiology 13 years ago. It's a teaching paper for radiologists, teaching them about what these resting state correlations are all about. And it's a nice paper. They do a good job. They take a region here in Broca's area. This is a patient with a brain tumor. And they show how there are correlations between Broca's on the left and Broca's on the right. And nowhere in the paper do they mention this, which is a crescent of correlated bold signal deep within the white matter, so it can't be a partial volume effect, but it just doesn't get mentioned in the paper. In other words, they weren't trying to teach that. This is actually a study that I started 26 years ago at Yale, and the people at Yale have continued it. We were looking at neonatal babies who were born prematurely, and the group there has followed those people for 20 years to look at their language development. So if you're born prematurely, maybe your language is delayed or something. And again, they talk about the language system, they talk about Broca's area here, Wernicke's area back here, back here. Nowhere do they mention this big strand of white matter that's showing correlated signal anywhere in the paper. And this is common. You can go into the literature and you can find lots of papers where there's evidence of signal in the white matter is not mentioned, and you know along the way they probably tried to get rid of it anyway. So it's you kind of can understand why it's been missed. I talked about methods of signal analysis. A very common method in the field of functional connectivity, resting state, is a technique known as independent component analysis. It's a statistical technique whereby you can tease out regions that show synchronized activity. So if you do this ICA analysis on the gray matter, you pick out these different regions as discrete components which are synchronized, and people talk about those as being nodes or parts of a network. Well, if you do the same analysis in white matter, you get very similar things. I mean, these are discrete synchronized regions using exactly the same processing method as for gray matter, and you get evidence of synchronized regions in the white matter, very similar to what you get in the cortex. And if you do the same in the spine, going back to the spine, if you do ICA on resting state signals in the spine, we published this. This shows 8 regions in the spine that show up as being highly synchronized. It includes the 2 dorsal horns, the 2 ventral horns, and then 3 regions in the middle. So this is the hubs, if you like, of networks within the gray matter, shown here. Well, if you do the same thing to the white matter, you get the same thing. You get similar things. You get 8 hubs of synchronized activity in the white matter of the spine, and you can assemble them around here. And if you overlay that with diffusion or anatomic sketches, these correspond very strongly with the major tracts within the spine that connect the brain to the nervous system. So it works in the spine too. So as an interim, let me summarize that white matter bold effects are robustly detectable, particularly during things like hypercapnia or in response to stimulations, just like in gray matter. There's also these temporal correlations in a resting state that you can detect by things like ICA, and you can pull out networks of correlated white matter areas. So some questions then arise. Is the bold effect in white matter different or the same as gray matter? And then what factors affect that signal in the white matter? And I can distinguish 2 types of influence. First of all, what I call biophysical. That is basic things intrinsic to the tissue, things like the composition, the neurovascular coupling, the microstructure. Then a second kind of factor might be how those white matter areas connect to the cortex. In other words, the signals in the white matter we know are related to neural activity in the cortex, so there are neurological factors that drive the magnitude of the signal in the white matter. And I won't have much time to talk about the second. I'll talk a little bit about the first group of samples. Now, we do a lot of work with animals. Professor Wu was surprised to learn, because I never talk about my animal work, but we do have animals. We have these nice little squirrel monkeys. This is one of their brains, and you can see they have nice big chunks of white matter. So you can study them in a resting state even though they're anesthetized. Now, if you anesthetize an animal and you look at the signal from the cortex, from the gray matter, you can vary the level of anesthesia. And so you can go from isoflurane at 0.5% to 0.8% to 1.25%, and you can see that the magnitude of these fluctuations goes down monotonically as you increase the level of anesthesia, and that's shown here in terms of the power spectrum. It goes from red down to blue to black. The normal explanation of that decrease is that you're decreasing baseline neural activity. Now, there could be other things happening because it's an anesthetized animal, et cetera, but the obvious conclusion is that the reason why these signals go down is because things are gradually less active as you increase anesthesia. So what happens in white matter? Again, over on the left, there's gray matter. This is white matter, and you can see again, the signal goes down monotonically as you increase the level of anesthesia. And that's sort of summarized here. This is muscle, where you wouldn't expect there to be any change. This is gray matter going down in a certain manner. That's white matter. So the fact that they parallel each other makes you think the explanation for why it changes in the Bold effect, in the white matter, is the same as for the cortex. That is, there's less activation, less activity within the white matter as you increase anesthesia. Now, I'm going to introduce 3 metrics of the Bold effect in the white matter, or actually in all brain. First of all, going back to that curve I showed when you had a transient task and you get some increase in signal, then a decrease, that's the hemodynamic response function, and it's produced by a very transient stimulus. And what I'm going to emphasize in a moment is that normally we kind of ignore this first part, but there is this negative dip at the start, and that's thought to be due to the short period during which there is an increase in neural activity, increase in metabolism, but the flow increase hasn't yet happened. So during that short time, there is more use of oxygen, so the level of deoxyhemoglobin actually increases temporarily. That means your signal goes down. And then the fresh blood comes in, washes that out, and you now have oxygenated blood, and the signal goes back up. So this has been very hard to find it's been predicted for 30 years. It's been very hard to find in the gray matter. Some people have reported it, but I'm putting it in there for completeness. And then there's this increase, and there's a peak, and it lasts for a certain time. Those low-level fluctuations kind of look like this if you blow them up, and you can talk about features of that, what you might call almost like a random noise signal, except it's not random. But you can talk about the fraction of amplitudes of these fluctuations within a certain range of frequencies. That's known as FALF. So remember the word FALF, right? It's the fractional amplitudes of low frequency fluctuations. I say FALF because to say that quickly is really difficult. But that's an important measure that people use to characterize those signals. And then you can simply take its Fourier transform, and you get some kind of spectrum, and that spectrum typically falls off kind of smoothly as 1 over f. Ignore these peaks for now. I'm going to come back to those, but typically they go down in a kind of smooth fashion. So you can talk about the spectrum, FALF, and the HRF. And this is the HRF, just to repeat that it has this negative dip, has this positive peak. The negative dip you don't see in the gray matter, but I'll show you that it's very prominent in the white matter. So if you compare, for example, across a whole brain, or actually across a whole, maybe 100 brains, you take all the white, all the gray matter, you can measure the HRF features. This is white matter versus gray matter. That's how they differ. There's a much bigger dip in the white. You can talk about the area under the dip. And the area under the dip this is a negative scale. It's much bigger in the white than in the gray. And all the other features are also different. So you can actually take each of these parameters, whether it's the peak height, the time to peak, the positive peak, the height of that peak, the width of that peak, how big is the negative dip. And you can see, if you look at the average values, that there's strong contrast between the gray and white matter. Now, the important point there is that means the HRF is different in gray and white. So if you do analysis that assumes a particular HRF, you will be wrong if you use the one for gray matter in the white matter. And that's another reason why I think people have missed that activation for a long time. My colleague, Kurt Schilling, did a very nice job. He took about 100 brains. He did tractography. He identified major tracts. And then he divided them into 20 sections. And each one is color-coded. So different color involves different distance along the tract. And if you look at a typical value, for example, there's a much bigger negative dip in the middle as you go into the middle of the tract than at the ends. And that's sort of summarized for one particular tract here, that one, whatever it's called, over on the, on the left. If you measure the area of the dip gets much bigger as you go into the brain and then comes back on the other side. And the time for that dip takes longer. So that's telling us something about the variation of the vasculature that's responding to neural activation, varying along the tract and also varying between tracts, which makes it much more complicated for when you do analysis. Now, we can look at the size, we can look at some of these features. We talked about FALF, we talked about the power spectrum. I'll show you now that they're highly related. They're actually all measuring the same thing. So here's the HRF, and it has this area of the negative dip. And what we did was we took a whole bunch of parcellations of the gray matter and then measured the streams between them, the white matter streams. And these are association fibers versus projection fibers shown in different colors. We can take that negative dip, we can take the power spectrum, and you'll notice, first of all, that in fact there is this peak in the power spectrum of the signal from the association fibers that's much weaker in the projection fibers, and which isn't there at all in gray matter. Turns out that that peak is in fact just reflecting the magnitude of this negative dip. If you plot one against the other, they are strongly related. So the negative dip and the peak in that power spectrum are measuring the same thing. And you can see also that they are different for these two types of fiber. In fact, that's what's shown here, that if you take association fibers versus projection fibers, and you count the magnitude of that peak in the power spectrum, is quite different in the association fibers. Now, to kind of explore the factors which affect this a little bit more, you can go to the literature, you can go to publicly available data, and you can find atlases of lots of different things. So, for example, there is an atlas of myelin water fraction, measured by taking a whole bunch of brains and doing a very special MR technique whereby you are sensitive only to the water in the myelin. There's other atlases that have cerebral blood volume, blood flow, CMRO2, oxygen extraction. We can do white matter bundles using diffusion. We can do DTI. And there's a technique known as NODI. I don't have time to explain it, but NODI gives you a measure of the neurite density. So it's a model of what happens in tissue and how much of it is actually due to neuronal content. And then we also have our own measures of HRF and FALF. So you have all these atlases, and you can merge them together. And then you can pick out each white matter tract and look at how do these different quantities correlate or covary across tracts. And this is a busy slide, but it's quite informative. So this, for example, shows you the amplitude of FALF, the amount of those low-frequency fluctuations, as a function of blood volume. And as you expect, it goes up the more blood you have. And it goes up very differently in projection fibers versus association fibers. If you look at how it depends on things like flow, it actually goes in the other direction. So the stronger the baseline flow, the less fluctuations you get. And so you can also infer things about the oxygen use being different in those two kinds of bundle. This is actually, I think, more interesting. How does that depend on myelin? So you can see there's a strong relationship between FALF and myelin content in association fibers, but almost no dependence at all in projection fibers. And even more interesting, this is my opinion, but even more interesting is that if you take the NODI data, and remember, NODI tells you about neurite density. So if we plot the neurite density here from NODI against FALF, and remember FALF is related to that negative dip and the power spectrum, you can see that there's a strong negative signal, strong negative dependence. Well, if this is neurites going this way, then going in that direction is everything in the brain other than neurites. So in the white matter, that means the glial cells. So you can take this as a surrogate measure of glial cell content. So the more glial cells you have, the bigger FALF. The more glial cells you have, the bigger the negative dip. the more glial cells you have, the bigger the power spectrum at a certain frequency. So this tells me that the bold effect in the white matter is not being driven by the same processes as you have in the cortex. In the cortex, the bold effect is driven by the energy demands of neurons. In the white matter, it's probably being driven by the energy demands of the astrocytes. So maybe we have here a method for looking at astrocyte function that is quite different to what we've been used to looking at with fMRI. Another little piece of proof of this, we found this in the literature. If you take the optic nerve where it comes out of the retina, the first few millimeters are not myelinated. Then it becomes very strongly myelinated. So if you also look at the mitochondrial density in the nerve, it goes down as you move along the optical nerve. So what we did was we went to the human connectome data. We found some 7 Tesla imagers sitting there, and they do a visual task. And that visual task we analyze, and it shows that it does cause visual activation in the cortex. And then we were able to find these slices in which you can actually see the optic nerve. And we were then able to measure the resting state signal along the optic nerve. And when you do that, you see, for example, when you have the task in the red area, in the visual cortex, yes, there's an evoked signal that goes up and down. But if you look in the resting state at the FALF values, then you can see that in the first few millimeters, there's a strong bold response, but further along there's none. So this again tells us that when things are fully myelinated, the signal goes away. So these big projection fibers with lots of myelin, there's no bold effect, but lots of other fibers in the brain which are partially myelinated or less myelinated, there's a strong signal, and that signal depends on not how many neurons you have, but how many glial cells. That's sort of what we think is the take-home message. So some interim conclusions. Bold signals are robustly detectable. The HRF is different, has a slower, more negative dip. They vary with baseline values like CMR02. Association fibers behave differently to projection fibers. But if you do the analysis just blind, everything you've always measured with gray matter and connectivity and stuff like that, you can find the same kinds of things in the white matter. Now, how is white matter bold signals related to gray matter? We showed a little bit about that. But what we'd like to show is that there's some relationship between overall neural function and these white matter fibers. So one thing we did, it's already a few years ago, you can take a whole bunch of brains, maybe 300 brains from the connectome database. You can identify parcels of the cortex. That's a standard technique. You can just download the parcels. You can also then go to another atlas and get the white matter fibers. Okay, so you can identify so many, in this case, a large number of fibers, in this case, a large number of cortical areas. You can then say, show me the relationship between the resting state signals in each of these fibers and each of these gray matter voxels. And this is the matrix that we produce from that, where these are cortical areas taken from an atlas. These are white matter tracts taken from a different atlas. And these are the values of the correlation coefficients between the resting state signals from each white matter tract and each cortical region. And it's been thresholded to get rid of a lot of stuff in the background. But the first thing you notice is this is not random. There's a structure here that shows things like some white matter tracts are very, very connected to the signal in lots of the brain, others much more sparsely so. So we call this a functional connectivity matrix. It was just our first chance to kind of integrate bold signals in white and gray. And the interesting thing is, if you compare that matrix between cognitively normal people and people with cognitive impairment, and also then those same normal people and people with full-blown Alzheimer's disease, and you say, show me the difference in those matrices, this is what you see. So what this says is that in Alzheimer's disease, there's a very strong degree of reduction in the white matter connectivity with the gray matter, and it gets worse as you progress along the spectrum. In fact, that's sort of shown here. This is the way in which just that total connectivity matrix. You just add up all those correlation coefficients. As you go from normal to mild cognitive impairment to full-blown Alzheimer's, it goes down monotonically. If you take the same subjects and you look at their behavioral measures, you can see that these are 5 different cognitive tests. They all go down the same way. And in fact, I would argue this is the first imaging measure that's ever shown a connection between the imaging measure and cognitive performance in this population. PET doesn't show it. Lots of other things don't show it. This actually does seem to reflect that integrity. Are we up to 50 yet? No, this was a later study where we went back and we looked at preclinical subjects. So these are subjects that show abnormality on PET. They have amyloid deposits in the brain, but they have not yet shown symptoms. And what we have here are a whole bunch of white matter tracts, and a whole bunch of white matter tracts, and a whole bunch of gray matter areas. And this shows you the connectivity in a resting state, the connectivity difference in a resting state between people with preclinical AD and normal subjects. So if they were all normal, this would be empty. But these are the connections that in a preclinical population are already showing major changes. And this is full-blown Alzheimer's dementia where lots of things have changed in addition. So this again becomes something that's quite provocative about can we characterize early changes in brain function in terms of white matter, gray matter connectivity from the bold signal and how that changes even before symptoms are evident. And that's, of course, what's shown here. This is actually the correlation of that reduced connectivity as it correlates with things like beta amyloid measured by PET or with cognitive behavior as measured by behavioral measures. And you can see, there is no difference between those patients with no symptoms, but as you get to Alzheimer's, there's a huge difference in these connectivities. So these plots are hard to interpret very quickly, but 3 minutes. But they are early evidence of the fact that this measure of white matter bold and its relationship to gray matter bold is telling us something that's not possible to get from other measures. So in my 3 minutes, I will read the conclusion slide, which is that bold signals are real and shouldn't be regressed out. They are similar but not the same as in gray matter. The HRF is different. They depend on microstructure, things like glial cells, and obviously it's going to depend on myelination and axonal size, a whole bunch of complicated things. Nonetheless, they show these correlations. Functional correlation matrices relate resting state signals, in particular tracts with gray matter volumes. These correlation matrices seem to change with things like cognitive decline. And I actually think we need to rethink some of our models of the brain to incorporate white matter in addition to what people are talking about with gray matter. And I'd just like to pay tribute to my colleagues who actually do the work. You'll notice there's 2 or 3 1, 2, 3, 4, 5 Chinese of the 8, which tells you something about the state of science in North America. Thank you very much.